Good afternoon, everyone. It's 4:46. I still see people trickling in from the break, but welcome to Bionano Genomics expert panel session, where we will be talking about optical genome mapping and its integration with next-generation sequencing for clinical research in leukemia and lymphoma. Now, this is an expert panel discussion, as you see 4 experts sitting here. But before we get there, I just wanted to orient everyone that may not know about optical genome mapping or OGM. So I only have one slide. For those of you that do not know and would like to know more, there are 8 or 10 posters outside, and please come visit our booth, or later talk to one of these experts who can tell you a little bit more. But in a nutshell, the overview is that if you have your cell lines, your blood samples, your bone marrow aspirates, the number one step, as we all know, is the isolation of ultra-high molecular weight DNA. We give you a very special kit to isolate this, and what do I mean by that? What I mean is hundreds of kilobase length molecules up to megabase length molecules, just to orient you over there. And then these ultra-high molecular weight DNA molecules are labeled with a fluorescent tag at a very unique 6-nucleotide motif along the entire genome. And what you see is that after that, these labeled DNA molecules are loaded onto a nanochannel array where they are linearized by electrophoretic mobility, and then they are imaged in our instrument, which is the next image that you see on this workflow. And when these images are captured, by virtue of the position of those labels, a digitized molecular barcode is made of your entire genome. It can be compared to your reference genome to reveal structural variants. What I do want you to pay attention to is this beautiful video playing in the middle. This is one of my favorite videos that I saw several years ago and fell in love with this technique. And I think that this is probably something that all the people in the audience can really resonate really well with, including our expert panel. So now let me introduce, it's really our honor to have 4 experts here today. We have Dr. Rashmi Kanagal-Shamanna from MD Anderson Cancer Center, who is sitting on who's the first person on the right-hand side, followed by Dr. Brynn Levy, whom you all probably know from Columbia University Medical Center, followed by Dr. Yassmine Akkari, and my apologies, she's at Nationwide Children's now, not at Legacy Health anymore. And then we have Dr. Ravindra Kolhe from Augusta University. You've seen a lot of presentations and posters from all of them over here. So I'm going to start this panel, and we're going to leave this slide on. My first question for the panel is, starting with Rashmi, is what are the gaps that you see in your clinical research testing when it comes to genetic or genomic testing?
Yeah, so my clinical and research interest is actually myeloid neoplasm, specifically myelodysplastic syndromes. And as all of you are probably aware, the treatment of any patient affected by myelodysplastic syndrome is based on risk stratification, for which you need the chromosomal abnormalities, the type, the number. Based on all of those, you risk stratify the patients. Conventionally, I mean, until now, even now, the gold standard to do this is conventional karyotype, right? But we know that not every case of MDS, like things like hypocellular MDS or cases which arise in the background of germline predisposition, we cannot culture those cells, so we cannot stratify them. And similarly, there are several abnormalities that are cryptic by conventional karyotyping, like KMT2A rearrangement, which is heavily prognostic here. And I work at MD Anderson, so there are tons of clinical trials happening, and there are several genomic-based clinical trials, precision clinical trials, for, just like KMT2A rearrangement, for which there's a menin inhibitor trial. So we have to take several steps to kind of interrogate these genes. On the other hand, we have the optical genome mapping, right, which can kind of, in an unbiased manner, generate all the results, the whole genome results, without going through those steps. So it's a matter of real, not just convenience. It's an advantage to have, especially when you work in such a scenario. And also, I find it really discrepant that using NGS, we are looking at a single nucleotide level at a high resolution. But for structural variants, we are kind of lagging behind to conventional cytogenetics. And I think this is a good opportunity for us to head forward and be at par with mutation analysis.
Thank you, Rashmi. Brynn?
So it's going to be easy because I think every one of us are going to agree with the person to their right. So yes, what Rashmi said, I will say, I'll make 2 points that I found particularly convincing or exciting about using OGM. One of them has become very apparent once, all Ravi, Yassmine, and Rashmi and many of my colleagues in the audience, we finished a multi-center trial looking at AML. And before the outstanding results and the increased diagnostic yield, one of the things that was apparent to me was that just looking at really good institutions across the United States, we don't operate the same. In other words, we have different fish panels. There's different clinician ordering practices. And that's not, unique to the US. It's pretty much, I think, you see this disparity across the world. And so with the introduction of OGM, you have some ability to eliminate disparities in how you offering or how you're practicing medicine and the types of tests that you're giving to your patients. In other words, they're pretty much going to get every fish panel on one shot, and you can really standardize the workflows. So that's the one aspect that I want to highlight. The other aspect that's also apparent is not only from the diagnostic side and increased diagnostic yield, but as Rashmi said, SVs are still being discovered. And we know based on current guidelines, you can look at high risk and low risk, and sometimes a patient that's in low risk just doesn't conform to that. They seem to be out of bounds from that, and the same for all the risk categories. And it's my suspicion that just these patients, when we're defining their risk stratification based on a carrier type or a couple of fish probes, we're really not getting the entire molecular picture of what's going on in these patients. And so I think we're entering a new era. I'm particularly excited about the discovery phase where we do something like optical genome mapping, and yes, we'll get the well-known things. But I think as we work together and put all our information together, we will start to discover the differentiators, why someone that's low risk is actually not low risk, because there's some other things that we never knew before. And I think that will be supplemented by NGS.
Absolutely. Thank you, Brynn. You make such excellent points, and Rashmi also. But I know that Yassmine, I'd like Yassmine to talk a little bit about, because you don't have firsthand experience with optical genome mapping. So can you talk a little bit about just a perspective of your laboratory workflow, the different techniques that you used, and what gaps you have seen when it has come to structural variation detection?
Yeah, no, I mean, first of all, thank you for having me, and I am going to agree with the people on my right. Alka, Rashmi, and Brynn. You better do the same. So from my perspective, I'm a big believer that emerging technologies have to stay with the times. And right now, our times are in crisis when it comes to staff shortage. I mean, we were in the panel yesterday, and we talked about something that everybody is really being challenged with. So one of the solutions in my mind is automation, right? Right now, I'm not saying that we should, decrease the expertise in conventional cytogenomics, but what we need to do is to go to technologies that can give you more with less. And one way that optical mapping can really kind of address these challenges is that you can get both copy number variations and structural variations in one workflow. A workflow that's actually a lot, more time efficient than other, previous technologies. It doesn't mean that it is not important to understand the dynamics of the genome, but what it means is that it gives you the same amount of results in a more efficient manner. The other thing is that when we think about diseases, and Rashmi and Brynn talked about the myeloid diseases, I come from an operation where we do everything. We do cancer, we do constitutional, and if we look at what test to use for what disease, there are certain mechanisms of disease that really dictate that we need to look for the CNVs and the SVs. And what, kind of better, more efficient way to do it, but to kind of see, okay, optical mapping can really give us those results for that particular disease.
So what was your reaction when you saw the results or when you saw this technique for the first time? I'm just curious,
Yeah, no, so I have a little bit of the imposter syndrome because I was invited to collaborate with Brynn's group because we had some patients that we wanted to see. And I actually, firsthand analyzed the results with Brynn, and it was really, really impressive how much more we saw. And what we saw that we thought by FISH is just a very kind of conventional fusion. When we looked at it molecularly by optical mapping, it wasn't what we thought it would be. And that would, potentially change outcome for this patient. So it's it's one of those, I'm one of those people who want that toy. But I don't have it yet, so it's stay tuned, maybe next year.
Thank you, Rashmi. So right before the break, we heard Dr. Nikhil Sejpal from Ravi's lab gave an excellent presentation, and then Ravi, you have so many tools in your toolbox that you play with. Yesterday, earlier today we heard your talk, and there are all of these posters. So can you comment a little bit about what do you use, what gaps you've seen, and where are we today?
Sure. Before I start, I want to say I agree with Alka, Rashmi, Brynn, and Yassmine. So I'd like to start with the middle name of my lab is esoteric. So anything which is esoteric comes to us in the entire state and the region. So my introduction to optical genome mapping was with COVID-19 host genome. Anything which was negative on whole genome sequencing, we started putting on optical genome mapping. There was a huge gap in understanding why some people are getting sick and why some are not. Whole genome was negative. Optical genome mapping provided the first solution in understanding that complexity. The consortium paper is out. It was amazing to understand why people are getting sick with the exact same viral load. Then we went and looked back and what are the real gaps in understanding this disease biology? So we went pretty much every disease type. We are looking at MRKH syndrome, 70 patients, whole exome sequencing negative, optical genome mapping is understanding. We are looking at transgender's genome, 60 patients or 60 individuals, both on both categories, whole exome sequencing is negative. We are looking at, can we understand with optical genome mapping? We're looking at prenatal, we're looking at postnatal, we're looking at, obviously, heme malignancies are a really interesting research perspective. We're looking at solid tumors, adult brain tumors. The research perspectives for optical genome mapping is out of the books. We have looked at pretty much everything in our lab which we are able to have or absolutely have negative from sequencing, karyotype, and FISH, and trying to figure out can optical genome mapping in itself can help us understand some of these issues. And we're going one step further and see how we can combine sequencing plus optical genome mapping to get a complete picture of understanding some of these diseases right at the time of their beginning and then follow them up.
Ravi, you really make a great point. So you just mentioned, all of these research projects that are going on that were negative by karyotyping or FISH or whole exome. So in terms of optical genome mapping and structural variants that you are actually finding, the cryptic ones, can you just tell the audience a little bit about some of just the variant classes and the types?
So I mean, they're all over we have 5 or 6 different variant classes which we have focused on optical genome mapping. And if you take any of these disease type, we could find difference in each and every variant class, especially if you're looking at the COVID-19 genome project, host genome project. We went into and proved by functional studies that these structural variants, which were very private events or true events, and by functional study we show that not only these events are true, but they're actually contributing towards the disease severity in COVID-19. We did the exact same thing with MRKH syndrome. We're looking at the very similar thing. I mean, uterine carcinosarcomas are such a complex disorders, malignancies, and having translocations by optical genome mapping is helping us understand some of these diseases. I think that's one of the major, I would say, translocation in oncology, and the other variant classes in non-oncology setting are helping us understand, because even whole genome sequencing is not able to get the kind of resolution for translocations the one we're getting on optical genome mapping. So translocations in oncology setting and the rest of the variant classes in the non-oncology setting pretty much are helping us understand some of these, or probably all of these issues we already have. I mean, one of the things I didn't mention was products of conception. That is such a challenging, for the last 30 years, when we went back and looked at our data, the most common cause was no growth, contamination, 46XX, metal cell contamination. We are not able to provide answers to our patients who come in purely because limitations of the technology. And then we have shown, and we recently published a paper on products of conception, that optical genome mapping can truly help. And in that cases, the non-translocation variants make a huge difference.
Mm-hmm. Thank you, Ravi. This is so nice just to, get this perspective from all of you. So obviously, Rashmi started with myelodysplastic syndrome and AML. We're looking at leukemias, lymphomas, which Brynn was talking about his research work. And then obviously, Yassmine talked about what we all deal with on a routine basis. You get any sample type coming into your research lab, and you want to find the answers. And then Ravi kind of wrapped it up with all the pillars of COVID-19. But I think what I saw or I heard, which I think all of you would agree with, is whether it's constitutional, whether it's prenatal research or postnatal research, pediatric undiagnosed diseases, or for that matter, heme malignancies, where structural variations play such a critical role, or even solid tumors, as Ravi was talking about the sarcomas and the glioblastomas. The structural variations have played a very important role, and the ability to find these at a high resolution are absolutely critical, as they all have just mentioned. So Rashmi, I'd like to go back to you, and congratulations on such an excellent paper that came out yesterday in leukemia, and you were talking about and alluding to the findings and how this impacted your research in terms of IPSS scoring or other scoring. Can you talk a little bit about that?
Sure, absolutely. I also want to mention that the project the inception of my introduction to the technology began during one of the CGC meetings, and it was happy to see the paper come out yesterday during the CGC meeting, like a full circle. So it's published in Leukemia yesterday. I encourage all of you to go back and read for the specific details, but just to give an overview, we had about 101 myelodysplastic syndrome patients, or it's myelodysplastic neoplasms, according to the WHO. So when we looked at the profile by cytogenetics, about 36% had normal karyotype. And using optical mapping, the proportion of normal karyotype dropped by 16%. So obviously, the karyotypes changed. There were a lot more complexity. Genomic complexity became apparent even in simple karyotypes. There were patients with chromothripsis that we were able to identify. There were a lot of biomarkers that we could tell even without doing FISH. For the paper, of course, we confirmed it with FISH, and optical genome mapping was very accurate in mapping those out. Things like TP53 gene, for example, in MDS, we know it's not just a mutation that matters anymore. We need biallelic alteration, so either a mutation with a deletion or a mutation with a copy-neutral loss of heterozygosity. So NGS doesn't cut it anymore. So you need NGS with optical genome mapping to classify based on the WHO subtype or the international classification subtype. So that's almost like a guideline now. So and optical genome mapping was able to do that.
So how did you tie the two data sets? You just mentioned, your NGS panel and...
Yeah.
Yeah,
So the NGS, we did, on all cases, we had the targeted NGS using 81 gene panel, and on all cases, same cases, we had the optical genome mapping. So we had a comprehensive mutation analysis. It was pretty deep coverage and optical genome mapping. So it was both structure variant profiling, mutation profiling combined. It was a pretty comprehensive analysis. So overall, I think together, NGS alone, about 10% did not have any mutations. And by karyotype, if we looked at the ones that did not have mutation, a significant number of them didn't have the karyotypic abnormality either. But if you look at them combined, 97% of all cases had some sort of a clonal genetic abnormality. Now, we know that doesn't equate to MDS, but in this case, combined with the morphologic dysplasia, it was diagnostic of MDS. But I think in this era, even detection of the clonal hematopoiesis is just as important because it has even bigger implications, right, in terms of cardiovascular mortality, et cetera. And the other point I wanted to, which is actually a very important, I should say, is the KMT2A partial tandem duplications. Based on the IPSS-M that just came out, I think 3 weeks. I mean, we have all these papers that came out this month. So, so the IPSS-M identifies KMT2A-PTD as one of the most adverse prognostic factors, along with the P53 biallelic alteration. And how many labs evaluate for this? No, I mean, microarray may be able to detect it, but pretty much NGS is not able to identify. FISH misses it all the time. And the IPSS-M stratification is out there, but the labs are not prepared to kind of implement it. I think optical genome mapping, we found 3% of the cases had them, which kind of matches with the publication. Their report also, they also found it in 3% of their patients, and it's beautifully, we were able to look at it. We actually identified it even before the paper came out, so we were kind of very interested in that.
You were scooped a little bit.
Yeah. And so that's fascinating, right? So using optical genome mapping together with targeted NGS, we will be able to stratify the patients even using this IPSS-M, and
Wow.
that's pretty, I think that's very important for the labs if they want to keep up with the published literature.
Thank you, Rashmi. Congratulations once again, and this is so important and absolutely amazing data that you have. Brynn, coming back to you, you talked about the AML consortium that you formed, and I want to say it was also conceived here at one of the CGC meetings. But what about your experience with exomes and genomes and your different research projects going on, and how do you think that ties in together?
So, yeah, so number one, like Ravi, the research that I'm using for OGM isn't restricted to heme or AML. We're going to be expanding this to all heme. We've got an ongoing, prenatal that's up and running, and we just completed some aspect of an ongoing pediatric side. I think it's going to be a very good synergy. We look at institutions that are doing whole genome sequencing, and certainly that can be the future at some stage. But the majority of diagnostic labs are not LabCorps and Stanfords and Columbias and Augustas, and the majority worldwide are smaller operations. And so things that are becoming a little bit more accessible to them are going to be things like sequencing. I mean, sequencing is slowly finding their way into smaller operations because it is becoming cheaper and cheaper. But I think it's going to be quite some time before whole genome sequencing.
Correct.
Just the infrastructure, the bioinformatic teams that you need, the resources So I think when we look at optical genome mapping and the decreasing costs with sequencing, I think it's a really good fit. Again, I see that there will be certain things you get from sequencing that you don't get from optical mapping, vice versa. But then there is also that small category that you're going to get from both combined. You know, one hit by sequence by NGS and some type of structural variant that you're going to miss.
Absolutely.
So I'm kind of excited about the combination of both.
Thank you, Brynn. That's an excellent perspective. Yassmine, you touched a little bit upon the workflow, and I know that you are really passionate about this shortage of technologists and directors, you know, expertise in the genomics field. So when you look at optical genome mapping, given that ABMGG also realized that the direction forward is LGG, right? So I'd like everybody to get your perspective on the workflow that you see, that you alluded to a little bit, but how do you see that in terms of analysis and interpretation and the tech shortage that you were talking about?
Yeah, and I'm gonna give a little bit of a disclaimer. So I do act as a reviewer for the COG ALL committee, and this is not the opinion of the COG. This is my personal opinion. But we are in a crisis mode where laboratories that submit COG data to the ALL committee are basically really struggling to find the abnormal cells. And how many, by a show of hands, culture cytogenetics for BALL? Okay, and how much effort do we put into looking at all the slides, making sure that we catch the abnormal clone? We just simply do not have the staff to do it anymore. This is a diagnostic, preparation of data, meaning that usually we have 90% blast, so we're not looking at minimal residual disease. The abnormality is there. And although I am not, like I said, I'm not speaking on behalf of COG, but in my mind, if we think a little bit about the future and think about the limited resources, the entry of the data into the COG database relies on the detection of structural variants as well as copy number variants, in addition to those entities of ALL that really require an SNVs. They're few and far between at this point in time for the ALL trials, and so it'd be really nice to kind of take a technology that is able to combine the molecular karyotyping in addition to a huge amount of fish panel into one technology that's affordable. The other thing that is it's important to I think all of us, when we think about healthcare globally, right? I'm coming from a third world country. I'm from Lebanon, and I have to tell you, in Lebanon, there is no NGS. It doesn't mean that cancer patients do not deserve to actually have precision medicine, right? And so we have to think about technologies that would capture the majority of the disease mechanism. And right now, we know for a lot of oncology studies, it is SNVs and CNVs and SVs. So a technology that is more, affordable by developing countries is extremely important to just global health.
Absolutely, I couldn't agree more. Because you bring such an important point, I just want to ask, how many people here are part of any DEI committee, ACMG, NSGC, AMP, by just a raise of hands? This was not by choice, but every speaker, and then me, the moderator, are really the poster child of diversity, equity, and inclusion here. So I think this itself is just absolutely phenomenal. I promise you this was not done by design. But thank you, Yassmine. This is a very important perspective, I think, for us to understand that the things that we talk about that are part of these professional society guidelines for anybody's clinical or translational research does not necessarily mean that there is access globally in terms of healthcare to these different countries. So thank you, Yassmine. That was a very, very pertinent and an important point. Ravi, coming back to you, we are talking about a research use only platform. So optical genome mapping is for research use only. And how do you see and how do you use all of the different tools in your toolbox with respect to different specimen types? You mentioned prenatal, postnatal, solid tumor, and cell lines, right, for these different projects that are going on. Can you comment a little bit on the specimen type, the workflow, and the different techniques in your lab?
Sure, I think we are reaching maybe 700, 800 samples in the last year and a half. We have 2 instruments which pretty much run 24/7. And we have went through a pretty rigorous process of going through and working with Bionano on all these research projects and then trying to get the best out of the key thing is the DNA isolation. So, and in each and every tumor type, not only cell type, but also the tissue type, that the breast tumor tends to be working different from uterine versus brain because you need nucleated cells with a certain number of presence to get a good quality of DNA in them. And we have worked out smaller things, and it has actually worked out pretty well. So you name each and every specimen or cell type. We had a protocol, modified, non-modified, had worked phenomenally well for the 700 or 800 samples in the last 1 year. And we know this is an RUO product, but I want everyone to take you back 10, 15 years when NGS was introduced, and we all took NGS. We started adapting it at the lab level. We did pretty rigorous CLIA validation, and we still pretty much each and every product of NGS in the clinical labs we are using is except maybe a couple, they are still RUO. I mean, if you look at any and every except the one I presented yesterday, they're all RUO products. And I think this is the path which pretty much every new technology will take unless you get some of these approved by FDA. So, and I think this is what the whole idea of lab medicine is. You're not going to get a product, you'll get a product at some point, but this is a brand new technology. You got the smartest people trying to figure out how we're going to use it in the constraint and the regulations given us by CLIA, as well as FDA, and make sure that we are doing a proper justice to these regulations we have. And we collectively come together. We have smaller user groups, we have smaller consortium trying to figure out right from how to validate them, how to work with specimen types, how to analyze them, how to report them, how to actually make clinical use of something like this. And as I said, we did this in the last 15 years with NGS, we did with microarrays. We will do something for single cell, we'll do for methylation. So anytime a new technology comes in, it spends a decent amount of time in the research space. Then it spends a decent amount of time in the validation phase. And I personally think we have done enough of OGM in those spaces. And I think truly it needs to be used collectively with everyone's effort for why we are here. I mean, to make sure that all these applications get really used for a better healthcare, our promise of better healthcare and precision medicine towards our patients.
Absolutely. Brynn, please.
So I think, again, we're in quite a unique position. Ravi appropriately refers us back, 10, 15 years. And if you think about when we started with the microarray era, it was kind of like the Wild West. Everyone was like out for themselves. And it still remains today. Some of the, the blowdown or the blowout from that is that there's still a huge divergence in reporting practices. Yeah. I mean, the way it's practiced is, I think the beauty is that we're taking the lessons over the last 15 years. Yeah. And we're in a really good position, like you saw from Adam Smith, if any of you listened to his talk, how all of us who are starting to do this are saying, it's going to be a lot better for us to work together. Let's have the mistakes together and see how we can learn from them, how we can, guide not only ourselves, but put down best practices. And I think we're doing it far earlier than, what we did for microarray.
Absolutely, Brynn, and Adam Smith is sitting there, and I want to acknowledge him too. He said something very apt. He said, turning subjectivity to objectivity. Is that what you said, Adam? Is that correct? He's nodding his head. So thank you so much for that. We have 10 minutes left. Yeah, Yassmine, go ahead.
Can I just, add just one more thing? I think what Adam is saying, I want to remind this audience that the genesis of this meeting was the Wild West of microarrays.
Yeah.
Right? And so, it is only appropriate that we are now talking about optical mapping in this audience.
Yeah, fantastic. We do have 10 minutes left, and so we are going to take audience questions. So please come to the mic, and if you have questions, the experts will answer.
Adam is coming.
Adam is an expert himself.
I don't know, can you see the online questions?
No, I cannot.
I think they can, okay.
We can do it.
No, I'll do that.
Are there any limitations? Because you made it sound wonderful. So, are there any things that you cannot do by combining NGS and the optical genome mapping?
So yes, the answer is yes and no. I mean, the thing is, well, what's your comparison, right? At this moment, we're comparing optical genome mapping to FISH and karyotype. In comparison to those two standard of care technologies for last, 50 plus years, there's a tremendous paradigm shift in what we are seeing in optical genome mapping. There is a number like 10,000 times more resolution than karyotype. So I would start with there. There are definitely limitations. I mean, obvious limitations of technology rather than limitations of what we want to do. And Alka can give a list of those, but what I'm more interested in is what we have missed in the last 50 years on karyotype and FISH, and how are we going to analyze that on optical genome mapping. And the major limitation I see is we can't take any SNVs on optical genome mapping. So I want to combine at least a targeted NGS panel plus optical genome mapping as a true combined assay where we are trying to at least investigate a lot of these oncology-based tumors.
Great question, Maxine. Brynn or Rashmi, if you want to add anything.
Yeah, I just wanted to add, to whatever Ravi said. I don't think one technology can encompass all types of genetic abnormalities, so it's kind of unreasonable to expect that. As Ravi mentioned, combining the SNV level to the larger chromosomal abnormalities, and I think from my experience, optical genome mapping seems to cover the aberrations that bridge those two. Mm-hmm. So that's, at least we are headed in the right direction.
Yeah. I will give you one, Maxine, FFPE. Optical genome mapping currently does not work on FFPE samples. So if you're looking for solid tumor, it has to be fresh or frozen. So So the question is that if you have a genome that has major complexity, how do you fit that with the information that you get from this technique?
I would go back to what Adam said. Complexity is complexity is complexity. Sometimes you just have to say it's complex.
Right? Great job.
No. So Maxine, I think we start with, when we started doing whole exome and even a bigger targeted panels for NGS, we would start with, let's say, 300 variants or 3,000 variants, and there's a magic word called filters. So you start filtering these and start making sense of those 3,000, and by the time I'm signing out these NGS cases for the last 5 years, you come to 15 post-filtering. So pretty much every genome is complex on OGM. So you put your filters in, you want to look at what's clinically relevant, and you can have these different compartments, what's clinically relevant versus on the research side. On the clinical relevance side, I think the consortium with Adam is leading or trying to figure out how to label these complex genomes. And after some point, it is just complex. And in that complexity, can we find something meaningful? And I think that's where the initial discussions on these consortium meetings were.
Thank you, Ravi. Maxine, I would encourage you to, if you want more details, talk after the session. Adam?
Yeah, thanks. I really wanted to touch back on Rashmi's point about KMT2A PTDs. And we actually have a poster, it's poster 63 for anybody who's interested. And what we did, well, a colleague of mine, Jose Mario Capuccici, who's been working on this problem from the molecular side for a long time, we have our NGS panel and an MLPA assay for detecting PTDs. And then I came to the party kind of late, and I said, wouldn't it be cool if we did OGM too? And we put them all together. We have about a dozen cases now. But what really stood out was, in fact, the fact that there's a lot of little things that can happen in KMT2A that cause problems. Like, you can have unbalanced KMT2A rearrangements. And you do MLPA, and MLP goes, that's a PTD. OGM goes, no, it's not. It's an unbalanced translocation. So there's like a lot of structural context that's super important. And I think that's something that OGM does really, really well, is put all of these things in structural context. And I agree with you, Rashmi, when you said that we're not ready for a lot of the stuff that came out in these guidelines. And I think, one of the ways to get there is actually using OGM. So, sorry, that's more of a comment.
Excellent comment. No, thank you, Adam. Our next question.
Hi, I feel like I need the T-shirt that says OMG.
OGM is here.
Yeah, but I do want to bring up the point of, it's very important to democratize cancer care and precision cancer care into LMIC, so low and middle income countries. And so I'm curious, we all know we can get molecular technologies for under the price of a cup of coffee in India, because that's what we did with COVID. So where do you see this being democratized? How do you see it being democratized?
That's a question for me. So I think I will answer this question. Maybe we should meet after the session is over. But as with everything else that we've seen, right, we heard a talk today where we were listening about how many years ago we heard the $1,000 genome, the $100 genome. So we are listening. We certainly understand that we need to work on the throughput and reducing the price, and we will certainly make our way over there.
Can I? Yeah. There's online questions.
Just to remind you.
Yeah. Can you take one?
But Ravi wanted to say
No.
something. Okay. There are quite a good number of online questions. I'll ask
We have 2 minutes left, so quickly.
Okay.
Yeah.
Dr. Kolhe, can you comment on the sample paucity and how OGM's limitation requiring high-quality DNA was overcome?
So I think we did a lot of workarounds, and we did a lot of experiments to figure out what would be the ideal cell count. And it's always, something as simple as the dead versus alive cells. When you're reviewing a cell line or when you want to get a fresh sample, you need a certain number of live samples to get a good quality of DNA. The storing mechanism, EDTA, works phenomenally best. Heparin does not work. So I think we're publishing a paper soon which will delineate all these different sample types we are using and the DNA isolation for them.
Thank you, Ravi. And I would just add that if the sample is frozen, whether it's your cell line, your tissue, your blood, or your bone marrow, optical genome mapping works very well. So just in a nutshell.
Okay.
And do you want to take another one?
Of course, questions are flying in now. We've got 40 seconds. So is there no enrichment step needed for OGM, and is the calling by de novo assembly to the reference genome?
There is no enrichment step that is needed. That is correct. There is no amplification, as you saw in the workflow that I showed you. And there are 2 different calling algorithms, the de novo assembly and a rare variant pipeline, depending on your application. And I encourage you to contact us later if you want more information. And we'll take one last question from our audience. It's yours.
Have you tried, or have you thought about, or do you think it would be useful to have a second color? To have an alternative restriction enzyme site that you're tagging, to mix them together, or to have a different one? Just what do you think about that, and have you tried it?
If I told you, then I would have to lock you, right, in a room somewhere.
Because it seems like you would.
So the answer to that is that we actually started off with 2 restriction enzymes before this protocol that I showed you has matured and evolved. Is there a two-color assay? There are numerous researchers that have worked on methylation and some other different applications using a two-color approach. We currently do not offer it, but can it be done? Yes, for specific applications. Thank you. So with that, I think this was one of the most engaging expert panel discussions that I've been part of for a very long time, and I'd like to thank all four of you, Rashmi, Brynn, Yassmine, and Ravi. Thank you so much, and thank you all for staying. This is one of my favorite meetings, the CGC, and I couldn't be more proud and honored. Thank you so much.
Thank you, Alka.